After years of research, the laboratory of Nicolas Rivron has found out how to induce stem cells to organize themselves and develop into an early embryo-like structure in a dish, all in the lab. This scientific breakthrough has truly opened up the black box of early pregnancy.
Surprisingly, we know very little about the first days of pregnancy. At that time, the embryo is extremely small. It's about the width of a hair, and it's hidden in the womb. It is floating inside the uterus. It contains less than 100 cells, and the cells from the outside are the stem cells that are going to form the placenta. Inside, there is a very small cluster of about 10 embryonic stem cells that are going to form the whole embryo. We can easily harvest, culture and multiply stem cells in the lab. We can do this for very early placental stem cells, very early embryonic stem cells, and they multiply while remaining locked in time. But in a natural embryo, those stem cells, they are together in 3 dimensions. And those stem cells, they are chatting, they are talking to each other, a language that we barely understand. What we did is to place them back together in a specific cocktail, and they started to converse. They started to spontaneously organize. So it's only mouse stem cells sprinkled with a handful of molecules that are self-organizing. This is like self-organizing life. Those embryo models are remarkably similar to mouse embryos, to the point that when transferred back into the uterus of a mouse, they implant and initiate a pregnancy. However, we clearly do not form a full mouse. These are just very useful models to research in the lab. So now we can form those model embryos in very large numbers, and we can study them in great details.
And through studying these early embryo models in the lab, we can achieve a great deal. We can finally precisely understand how adult diseases start within the embryo, thus enabling us to prevent and reverse their appearance. We will also be able to discover new medicines without animal experiments. For instance, medicines to help couples who experience difficulties when it comes to having a child. All of this begins with the same thing, understanding the communication between stem cells.
What we observed is that the embryonic cells are the chatty ones here. They are the ones that are instructing the placental stem cells to multiply, to organize, and also to implant into the uterus. So this stem cell conversation that we seized opened very new perspective to research in the lab the problems of infertility or pregnancy failure. For example, 2/3 of the in vitro fertilization treatments are currently failing, and they are failing at the time of implantation in the uterus. And we just don't know why. Using those model embryos, we can test and develop new molecules for the embryo to take a perfect path for a successful and a healthy pregnancy. These new molecules that we could find might become the medicines of the future.